All right. Welcome to class, guys.
How's it going? Good yourself?
Good, good. We will be covering how cells can divide and grow into new cells today. So this is called cell division. And so I'm going to give you guys some packets, as well as I'd like to ask you to please get on Google Classroom because we have some links that we'll be working with during the lesson. So we begin by introducing a question. I have a question for all you guys. When you get a cut and your skin heals, how did your skin cells come back? What do you guys think?
Do they just come together? Do they form new cells?
Right. I think everyone understands that if there's a cut, right, that there's going to be a gap where their cells used to be. We learned that all human tissues are made of cells. That includes skin, liver, bones, brain, all of your tissues are made of cells. And so if you get a cut, that means that there is a gap where there are no more cells. However, you're going to need to replace those cells. If you need to grow, heal, or replace those cells, it means yes, you need more cells. The way the body makes more cells is through a process called cell division. This is essentially a cell doubling. You start with one cell, and then you end up with two identical cells. And this process is very important for a number of reasons. Your body can do this in order to grow. All of you in this room used to be the size of a small baby, but now you're much, much larger. For that to have happened, we needed to have new cells formed. And as we learned in the first cell unit, all cells come from
an old cell?
Old cells, that's absolutely right. And so this process of having new cells come from old cells is called cell division. Today we're going to be talking about cell division specifically for somatic cells. And that means your regular body cells. That's pretty much all of your cells except your sex cells, which means sperm and eggs. They divide differently, and we're going to be covering that another day. So as we can see, we start with a cell, and then we end up with two identical cells. This isn't reproduction when we're talking about things like humans and multicellular life. This is simply making a copy of the cell. So the cell cycle is essentially how cell division works. It's every process of the cell cycle. It's every process of cell division put into one loop. It's called a cycle because your cells are pretty much constantly dividing. There are some cells that are not dividing, but for the most part, even though it's not very quick, all of the cells in your body are dividing. The first stage of the cell cycle is called interphase. And a good way to just think of interphase is this is just when the cells are doing their thing. Their skin cells are acting like skin cells, immune cells are acting like immune cells, and they're not actively in the process of dividing. However, they are preparing to divide. Interphase, while they're not dividing, is the place where DNA is replicated. And we're going to talk about why we have to replicate DNA in order to do cell division. But what I want you guys to look at here is how interphase makes up a vast majority of the lifespan of a cell. The cell is usually an interphase. And so when we've looked at cells under the microscope and we see that one kind of floppy, fried egg-shaped type of animal cell that has an intact nucleus, it's in interphase. Most cells in your body right now are in interphase. So interphase, again, is just sort of the default of what a cell is doing, and it's different than mitosis, or active cell division, which we're going to cover today. So the only thing you have to know about interphase is that it's all of the parts of mitosis, it's all of the parts of the cell cycle that is not mitosis, and it's the phase of the cell cycle where DNA is replicated. But why would DNA have to be replicated? Let's talk about that. If each of your cells has one complete copy of DNA, and one alone, how could it possibly divide into two cells? Anyone want to guess? How come a cell with only one copy of DNA could divide into two. What does it have to do first before it divides into two? You know, let me actually ask you guys this. What's the organelle in the cell that has DNA?
The nucleus.
Yeah, the nucleus. Most people understand the nucleus, right? The nucleus is the center of the cell. It's the brain of the cell. It contains all the instructions for the cell to perform its functions. And so, if you have one nucleus and one complete copy of DNA, what do you have to do before you can divide into two cells? Yeah.
You have to make copies of your DNA.
Right, that's absolutely right. And this is the first most important thing we have to focus on today. You have to make a copy of your DNA before the cells can divide. Because, and focus real closely on this, if we divide a cell, that means we have to also divide its nucleus. And this is really the most complicated part of cell division. To some extent, you could take a cell and sort of, you could imagine that you could just take half of the contents of one of the cell and half of the contents of the cell and separate it into two membranes, and the cell would work. That actually would be true, except for the fact that it needs to have its own nucleus, and it needs to have one complete DNA genome. And so before a cell can divide, it needs to replicate its DNA. If one cell divides into two, we first have to have two copies of DNA. And this process happens during interphase. Just to remind you guys, that is the step that occurs before cell division. And this is where DNA is replicated. Now, the thing is, though, that during mitosis, as you guys are going to see, right, and it's okay if you don't fully understand this right now, DNA has to condense into something called chromosomes. How many people here have heard of chromosomes? Right, quite a few people have. And chromosomes, what they are very tightly packed form of DNA. I think a very good way to look at and consider it is to think about something like, say, a charging cable. DNA, as we talked about, is a string. It's a polymer. It's a long rope of connected nucleotides. And that's actually the way it is usually in the cell. In the nucleus, DNA is usually something like this. It's called chromatin. And chromatin is not very tightly wound up. It's not very condensed. It's kind of loose. And the reason is because the DNA is in use, right? Proteins have to access the DNA needs to be used by the cell, and so it's in this sort of loose, tangly form. But as you might imagine, because we need to have exactly one half of the DNA go into each of the two resulting cells, called daughter cells, in cell division, we have to actually package this DNA up temporarily to get it to move to each cell. And so if a chromatin, which is the form of DNA that cells are normally have in their nucleus during interphase, chromosomes are like this. It's a very condensed, tightly wound structure that we can call essentially its own piece of DNA. So this is a chromosome right here, but this is chromatin. So during mitosis, in fact, the first step of mitosis is this condensation or packaging of DNA into chromosomes. Does anybody remember, has anybody ever heard how many chromosomes humans have? Yeah.
46?
46, that's correct. And we're going to learn more about chromosome number as we move on, but today we just want to talk about mitosis, so we can understand it as a process and use that as a way to understand chromosome number and mitosis. So, as you said correctly, humans have 46 pairs of 46 chromosomes, but they come in 23 different flavors. So even though you have 46, you have 23 pairs. You have one that you get from your mom and one that you get from your dad of each of the 23 flavors. So we can look at this picture for a second and we see that there's 23 different types of chromosomes. You have 2 number 7s, 2 number 10s, 2 number 18s, one that you get from your mom and one that you get from your dad. So one complete set of DNA is 46 chromosomes. One complete set of DNA is 23 pairs. And when you said that there's 46 chromosomes in human cells, that's true for somatic or body cells, the type of cells that we're talking about today. In another lesson, we're going to talk about how gametes, or sex cells, like sperm and eggs, have to have half of this number of chromosomes, or 23. So now we know what chromosomes are. We understand that cells divide, and we understand that before they divide, they have to replicate their DNA and condense them into chromosomes. Now, before we move on, I just want to ask one thing. If each of your somatic cells starts with 46 chromosomes, how many chromosomes do each of the cells have after mitosis, after cell division?
23.
So, a lot of students would say 23, but we have to remember that mitosis begins, right? It doesn't begin until you've doubled the amount of DNA. When you do mitosis, you turn one cell into 2 identical copies of it. And those identical copies therefore have to have the exact same number of chromosomes. So actually, there's 46 chromosomes in the cells after mitosis as well. And think about that for a second. If that wasn't true, you wouldn't really be able to heal a wound because after a few divisions, you'd run out of DNA. So mitosis retains the exact same number of chromosomes in the beginning and the end. Okay, now we're starting to look at mitosis and how mitosis works. Mitosis is specifically the part of the cell cycle where the active form of cell division actually occurs. The nucleus divides into 2, moves to 2 sides of the cell, and then the membrane splits off, forming 2 identical cells. We start with a cell like this, and we end up with 2 cells like this. That's cell division. So mitosis happens in 4 phases that we're going to talk about today. And we want to get ready to be able to do a lab where we're able to identify each of the different phases of mitosis. So first of all, let me ask an important question. Is interphase part of mitosis?
Yes.
Right, a lot of students might think so, but interphase is actually what happens before mitosis. And that's why I started by separating interphase from mitosis and cytokinesis. Interphase is just the normal default state of a cell. It is a place where you're preparing for mitosis by replicating your DNA, but it's not actually mitosis. Mitosis begins with a first phase called prophase, right here. Everybody on Google Classroom, you should be following along on looking at this picture so you can look at it closely. What's unique about prophase? How could I tell that a cell is in prophase and has actually started mitosis? Yeah.
In the picture, it shows that the blue stuff is more condensed than in the interphase. So
Right.
I'm guessing those are the chromosomes. You can see the chromosomes.
That's right. And to clarify for everyone else, what's drawn in blue is DNA. So we can see in interphase the DNA is tangled. It's loose. It's chromatin, right? Just like the example I used with the charging cable. We know that mitosis starts when the chromosomes actually condense into these units which are easy to move and separate into 2 different cells. So once we can see that this blue stuff, or DNA, has condensed into chromosomes, that's how we know that prophase has started. We can also see that the nucleus has started to degrade, but we still do see a nucleus. What's unique about metaphase that we can get from the picture?
It looks like the lines are connected to the chromatin.
Excellent. So these lines are something that we might remember from the organelles section. But if we don't, we can review it right now. These lines are called spindle fibers. As you might imagine, I've kept telling you that the chromosomes are separated. and brought to two different sides so that they can be part of two different cells. But something then has to pull on the chromosomes. What pulls on the chromosomes are called spindle fibers, and that's what these black lines are. They come out of and emanate from centrioles or centrosomes. And while we went over centrioles and centrosomes in the cell organelle section, it's something that's worth going over again. Centrioles and centrosomes are easy to remember. You can see that that's where the spindle fibers are coming from. So I like to describe the centrioles and centrosomes like Spider-Man's web shooters or a can of silly string. This is where the spindle fibers come from, right? This is what shoots them out and also pulls them back. Now, after metaphase, we have anaphase. What's the difference between metaphase and anaphase? In anaphase,
In the picture, it's showing the chromatin being separated.
yeah, the chromosomes being separated. That's absolutely correct. In metaphase, the chromosomes were aligned in the middle of the cell that's about to divide. And then now we see that in anaphase, the chromosomes got pulled apart. That's the difference between metaphase and anaphase. So it's pulling apart the chromosomes so they can go to two different ends of the cell, or poles of the cell. And that's what anaphase is. And now, after anaphase, after the chromosomes have been pulled to each side of the cell, we see that they are in telophase. And how could I tell that we're back in telophase?
The picture looks like two eyes that are connected with glasses.
Right, so what are these two things that have formed? It contains the blue stuff, which is DNA. And what organelle contains it?
The nucleus.
That's right. So in telophase, we can see one cell with two nuclei. The cell hasn't completely divided. We can see that the cytoplasm and the cell membrane are still shared. But that's happening right now. So we can tell that telophase has happened because it sort of looks like the chromosomes from interphase, but we can see that it's still one cell with two nuclei. Now, those are the 4 phases of mitosis. And then the last one, which we're going to cover later, is just called cytokinesis, and that's when the membrane actually divides. But mitosis is this process of dissolving the nucleus in prophase, condensing the DNA into chromosomes, aligning them in the middle, separating them, and then making 2 nuclei, right? You need 2 nucleuses to have 2 cells. And so that's why mitosis is so critical. It really happens like this too. I always want students to realize that this is really something pretty amazing. It's not just something that you can see in a cartoon, or this is some image that kind of estimates what it looks like. In this video, you'll see that they've dyed the DNA red, and they've dyed the spindle fibers green. And you'll be able to see that each of these steps, each of these phases, from prophase to metaphase to anaphase to telophase, really happens. And this is happening in your body multiple times a day. You can see how the chromosomes fell apart. The nucleus fell apart, and the chromosomes aligned in the middle. And then they separate. What phase do we think the cells that we're in mitosis right now in this picture? Can anyone guess? You're welcome to look at your notes for this.
Anaphase?
So it's not quite anaphase, but I can see why you thought that. It does have spindle fibers, and they are connected to the chromosomes. But have the chromosomes been ripped apart yet? No, right? So what phase is this?
Metaphase?
Excellent. This is metaphase. Metaphase is also called metaphase because it's in the middle. And meta, the root word meta, means middle. So it's just that the chromosomes are centered in the middle, and they haven't yet been pulled apart. So with my fingers, I can show you guys a little trick. This is metaphase. This is anaphase. This is metaphase. This is anaphase. common question students have is, how long does this process take? Is it happening this fast in the body? Not quite. These are sped up images. And while it varies a lot, cell division and its speed really varies depending on the function of this tissue. So your skin is dividing on a basis that only takes days or weeks. But the tissues in your brain can take years to divide, sometimes not even divide at all. And so this is a process that takes quite a long time. Cells don't divide very fast unless, as we're going to talk about, they're either bacterial cells, which can divide very quickly, or our cells can divide very quickly too, as fast as once every 24 hours if you have a certain disease. Now, so a key question that I get pretty much every year is, how do the cells know when to stop dividing? As you might imagine, we started with the principle of the idea that we have a cut, right? And if you have a cut and the cells get replaced, you don't continue to see the cells grow and grow. You don't get a huge amount of skin that keeps growing. When you grew from the size of a baby to, when I grew from the size of a baby to my size now as an adult, I'm not going to be growing anymore. At some point, cell division has to know when to stop. So, if cell division does not stop, this causes a disease called cancer. And cancer is a disease of excess cell division. Cancer is something which occurs because some cells don't know when to stop dividing. Normally, your cells do know when to stop dividing, for many reasons. For example, they can sense that there are other cells around them. This is called contact inhibition. When they make contact with the cells around them, they know that they don't need to grow anymore. But some cells, because of mutations or changes in their DNA, lose the ability to detect when they should stop dividing. How many people here have heard of cancer? And how many people here have had a family member or a friend with cancer? Cancer is a very difficult disease because, unlike diseases like COVID or the flu, there's nothing to target or kill. There's no external invading pathogen. There's no bacteria that we can kill with antibiotics. Because cancer is a disease where your own cells are dividing too frequently, it is your own cells that are the enemy. That makes it very difficult to make medicines to prevent or kill cancer. Okay, so all of this today, right? Let's go ahead and look at a little mini quiz to make sure that we are on the right track. Everybody, please go ahead and move to this slide. You'll see it after mini quiz. And please, in your notes, go ahead and write the answer to these 5 questions. It starts with something simple. What are chromosomes, and in what stage of the cell cycle are they formed? And I ask you a few questions about mitosis. Please take a few minutes to do that. So at this point, the students would take a few minutes to do that, and I would go ahead and use that information to go over another topic or anything like that in order to address anything that didn't stick. Since it's the mini lesson, I'm going to skip over actually going over the assessment here. Now, what I do want to do is get ready for tomorrow. The reason that we were learning mitosis today is so that we can apply that information for a lab activity where we're going to identify different cells that are in different stages of mitosis. What I'd like everyone to do now is please go ahead and click this link. It's on Google Classroom, but it's also on the PowerPoint presentation as well. And it's going to bring us to a virtual microscope. Tomorrow, we're going to be using the microscope, and we're going to be looking at onion root tip cells. Now, what do you think is unique about the tip of a root? Why do you think we picked that to learn about cell division? Anyone want to guess? Yeah.
Is that where it's growing? The plant is growing?
Excellent. You guys might have remembered from the plant part section that roots grow from the tip, right? So they get longer and longer and longer, right, from the tip, which means they're dividing at the tip. So I'd like everyone to go into this virtual microscope right here and press Explore. Now, when you press Explore, you'll be able to press this box right here and go to Plant Slides, and you're going to pick Onion Root. Everyone, please make sure we're on that right now. Now, all I want to do is go over the first couple of steps on how to bring a slide like this into focus. So right now, we have the slide, but it looks like this. What can I do to make it clearer? What's the first thing I should do to make it clearer? Joe?
I have no idea.
Do you remember,
To
Alan?
make it clearer?
Yeah, what's the first step we have to do?
Clean it?
That's actually not a bad idea to clean the lenses, but I'll have cleaned the lenses for you. Let's say you put the slide underneath the microscope and you see it looks blurry.
Are you like slowly adjust the course focus to up and down to see if it becomes more clear?
Excellent. We got to use the coarse focus first, guys, because it moves, as you can see, the stage up and down a lot. We only use the coarse focus when it's at the lowest magnification, 4x. So when we do our standard procedure and we start at 4x, we have to start by using coarse focus. This is the last time we'll use coarse focus. We're not allowed to use it on the higher mags. Then we can go up to a higher magnification, like 10x. As we go up in the higher magnification, don't forget to turn the light up, because as you go up in magnification, it's going to get darker. Slowly, we'll keep on making fine adjustments and adjustments until we get to 40x. At that point, we can clearly see each of these cells. These onion root tip cells, we can tell they're plant cells because they're boxy. Anyone remember what's the organelle that makes plant cells look boxy and rigid?
The cell wall?
Absolutely, the cell wall. So you can see that each of these are an individual cell, and each of these have a nucleus in them that's stained blue. Your job is going to be, and let's go ahead and look at the lab I just passed out, is going to be tomorrow that we're going to be looking at these cells. We're going to see that they're going to be in all different parts of the cell cycle. Some are going to be in interphase and not even in mitosis. Some of them are going to be in prophase, metaphase, anaphase, and telophase. And so at that point, we're going to be able to use this chart to identify what percentage of what cells are in which phase of mitosis. So all I want you guys to do right now for the last couple of minutes is go ahead and use this to identify cells that are in a particular phase. For example, can anyone tell me what phase this cell is in?
Telophase or anaphase?
That's a really, really good nuanced way to answer it. It seems like it could be in telophase or anaphase. But I'll tell you, this one is anaphase because we haven't seen the nucleuses reform yet. So this is anaphase because the chromosomes have separated. Remember the finger trick. Metaphase, anaphase. Good. Let's do one more, just one more. What about a cell like this? You're going to see cells like this all the time, in fact. This cell is in what phase? Cells like this one, or this one, or this one.
Interphase?
Right. And why did you say interphase?
Because there's one nucleus and I can't see the little blue squigglies in there.
Excellent. These are interphase. Most of the cells you'll find are in interphase. And that's because the chromosome is intact and we don't see any chromosomes. And it's just one cell. Perfect. This is what we're going to be doing tomorrow. So, in order to finish up, what I'm going to do is I'm going to go ahead and ask you guys to finish with an exit ticket. If you don't finish the exit ticket in class, you can finish it for homework, but we click it right there. And it's going to be just some short answers on what we've learned today. We're going to use the last few minutes of class to help answer any questions. And I'm going to come around if anybody has had a problem accessing that microscope, the virtual microscope. Please practice it for homework today because tomorrow you're going to be using it for real. Thank you. Awesome. Yeah.
That's good.
All right.